So this lecture is about what genomic data science is. So I'm going to try to quickly introduce the major disciplines that comprise genomic data science and tell you what we consider to be part of it. So genomic data science is in the intersection of biology, statistics, and computer science. And we're calling it genomic data science, but it actually goes by many other names, some of which you might have heard, such as computational genomics, computational biology, bioinformatics, or statistical genomics. Regardless of which name you use, the activities that genomic data scientists do all sort of fall into these into similar categories. So what do we do in genomic data science? We start with let's talk about studying humans. We start with some subjects. By the way, it doesn't have to be humans. It could be mice or model organisms. But let's say we're talking about humans. We'd want to go and collect some samples from those humans. So those could just be, if we're looking at normal development, we might just collect some skin cells from normal people. And then prepare those samples in the lab, send them for sequencing. The sequencer then generates enormous amounts of data that we have to do something with. So what we typically will do when we're looking at humans is we'll take those sequences, which are very short fragments. We call those reads because you're reading the DNA. So I'll use the term reads, and others will in this course as well, to mean a little bit of sequence that's come off a sequencer, which is part of someone's genome. And we take those reads and we align them to the reference human genome, and there is one reference genome for now, which represents, for now, sort of average Northern European male. In the near future, we're probably going to have many other reference genomes as well. But in any case, we align these reads to the reference genome, and that tells us how they differ from the reference genome. It also lets us kind of pile them all up on one another and see what sort of differences are even within the genome. Now remember, for every genome of a person, you actually have 2 copies of every chromosome, and that's because you've got one copy from mom and one from dad. So one of the things we study, for example, at this phase is how do your copies of each gene differ between the 2 copies that you have. And then once we've done some kind of analysis, we may deposit that data in a public database, such as one of the databases at NCBI, and we apply many other types of analyses to these data sets to make more biological conclusions about what's going on. So that's sort of very, very broadly speaking what genomic data science is. So we start typically with experimental design. So if you're going to do genomics, you have to first think about, well, I have a scientific question I want to answer, and I have to decide, well, how much data do I need? How many subjects do I need? What kind of data do I get? I've been talking about sequence data so far, but actually there's various modifications we can do, some new technologies that go by names like ChIP-seq and methyl-seq to let you study other things about the genome as well. So the first thing you do is you come up with an experimental design which, if everything goes well, you hope will allow you to answer those questions. And that's really critical. If you don't, these experiments, even though we're generating a lot of data far more cheaply than before, it still is expensive and time consuming to do these experiments. And you don't want to reach the end of your experiment and discover that you simply don't have the right data to answer your question. So once you've decided on your experimental design, you generate your data. Then you'll take those big data sets, and as I was saying a few minutes ago, the first thing you'll typically do is align them to the reference genome and find out how they differ from that genome, and assemble them together in some form. So that might mean that you're looking at, say, if you've captured RNA from the cells, when you align them to the genome, you can see for every gene in the genome how much of that gene was present. So that's the kind of thing that you'd be doing at the part where you're aligning and assembling things. You might be assembling the genes that were expressed in a set of cells or tissue that you were studying. So another big important step in this process is preprocessing and normalization of the data. Now because these data sets are very big, there's various types of biases that can come out. And this is sort of the law of large numbers comes into play a lot here. Even all of the technology that we're talking about, even though I'm talking about it like it's this very nice, clean technology, it's not perfect. So the sequencing machine itself, it makes mistakes. The process of collecting the data introduces biases. We might collect more of some tissues than others. We might collect some genes might be represented at higher levels because of biases, not because of true biological differences. The sequencing machines themselves sometimes have errors that are not random and they're hard to identify. They have other errors that are random, and we can identify those and get rid of those. So we basically want to take these big data sets and apply some computational and statistical methods to correct as best we can any kinds of systematic errors or any sorts of bias that are in that data before we then move on to the next steps. So once we've got the data normalize, preprocess, and normalize, we then apply a variety of techniques that come from fields within statistics, within computer science, and within biology to make our conclusions. So for example, in statistics and machine learning, scientists develop a wide variety of techniques, some of which we'll talk about later in this course, to go from this pre-processed normalized data to scientific conclusions. So in order to then take that normalized data and make these conclusions, one of the things that people within the field of genomic data science themselves do is develop software. So when we have an experiment, and I'll just take one as an example, one that I'm very familiar with, is RNA-seq. So RNA-seq, which we'll talk about probably time and again in this course, is a set of experiments, an experimental protocol where you capture just the genes that are being turned on in a set of cells, and you measure those by sequencing. And this is an experimental paradigm that's very popular. You can use it to study cancer, you can use it to study development, you can use it to study evolution, and hundreds or thousands of different kinds of conditions. And because of that, the experimental protocol for doing the RNA-seq part is more or less standardized. That means that we can design software that will, in a more or less standardized way, go from these raw sequence data to something which is more amenable to making biological conclusions. What you really want when you're doing RNA sequencing is you would like to know, okay, I had my samples. I want to know in each sample what genes were turned on and what levels were those genes present in. So there are software packages to do that that are developed by people in genomic data science. And software development, of course, requires not just getting the code to work one time, but also documenting it, making sure it works for lots of different cases. So there's a lot of software engineering kind of principles and algorithmic principles that are important in doing that sort of development. So another thing that we study in genomic data science is another broad category of questions, or population genomics. So I've mostly been talking about individual sorts of questions like what causes a cancer to be a cancer. But we can study other things too, such as we can look at a population and ask what makes a particular group of people more susceptible to a particular type of disease, or why do some people have resistance to a particular type of disease? So, or why do some people have certain traits in their population? So population genomics refers to looking at, rather than looking at one person at a time or small groups, looking at whole populations and studying their genomes to see how they differ and how those differences lead to recognizable differences in the people in those populations. So another way to characterize genomics and to characterize some work in genomic data science is when we collect experiments of different types and pull them all together. So you might call that integrated genomics. You could also call it systems biology, another popular term. But that refers to computational and statistical activities where we take data that comes from sequencing experiments, say, of different types, as well as other kinds of measurements. There might be measurements on what we call the proteome, measurements separate from the DNA, and integrating those all together to make biological conclusions. So we would call that integrative genomics.